お待たせいたしました。本日はご視聴承ります旨、ありがとうございます。司会を務めます、イスラエル大使館経済部、綾尾健二と申します。よろしくお願いいたします。Thank you all for joining us today. This is Kenji from your Israeli Economic and Trade Mission. 皆様へ一点お願いがございます。各講演者へのご質問は、Zoom 画面下の Q&A をご活用いただき、英語にて、また講演者の名前も記載願います。なお、すべてのご質問にお答えできない折には何卒ご了承願います。Please utilize the Q&A function and also indicate the presenter whom you are addressing. We will try our utmost in responding in timely manner. では、まずはじめに、本ウェビナー開催におきましてご支援いただいております、一般社団法人再生医療イノベーションフォーラムより、国際委員長、野村昌行様からご挨拶を受けてまいります。We are privileged to have the opening remark from Dr. Max Nomura, Chair, International Affairs Committee of the FIRM, the Forum for Innovative Regenerative Medicine. 野村様、お願いいたします。
はい。じゃあ、まず最初に日本語でご挨拶させてあげます。こんにちは。再生医療イノベーションフォーラムの国際委員長の野村と申します。皆様、本日お忙しい中、本ウェビナーにご参加いただきまして、誠にありがとうございます。僭越ながら一言だけ開会に際してご挨拶をさせていただきます。今日はバイオコンバージェンスと再生医療、日本・イスラエルにおける競争の機会と題して、イスラエルの再生医療における動向や、当該領域で注目すべきスタートアップ3社のご紹介、そしてウェビナー最後のパートでは、イスラエル企業と日本企業の事業提携に関する多くの可能性についてのパネルディスカッションが予定されております。で、このバイオコンバージェンスというアプローチは、バイオテクノロジーと工学、AI、物理学、計算科学、ナノテクノロジー、それに材料科学とか、あるいは先端的な遺伝子工学などを統合化することで、健康とか医療、それに農業、エネルギーの領域まで含めてですね、そういう分野における課題の解決、あるいはその糸口を見つけ出すアプローチということで、イスラエルはこれを次の経済成長エンジンとして、経済成長エンジンとして位置づけているということです。で、昔からイスラエルには強い起業家マインドが浸透していることはご存知の通りですけれども、ヘルスケア領域においても革新的なソリューションを提供するイスラエルのベンチャーは、その存在感はですね、トップクラスです。で、本日も日本でいうところの再生医療の枠には収まらないイノベーションが紹介されます。実行力、スピード感など、日本の我々も学ぶ点が多く、本日をきっかけにしてさまざまな競争の機会が生まれたらと、幸いだと思っています。皆様にとって有意義な会になることを記念いたしましてご挨拶とさせていただきます。Hello everyone. I'm Max Momura from ISCS Corporation, Japanese chemical and material company, and work as the chair of International Affairs Committee of FIRM. Thank you so much for joining today's webinar. FIRM stands for the Forum for Innovative Regenerative Medicine. It has been established in 2011. Our mission is to accelerate industrialization of regenerative medicine. We have more than 200 member companies. It consists of pharmaceutical company, chemical and material company, and machinery device company, logistics, insurance, and consulting companies in Japan. FIRM have global partnership by MOU in the field of regenerative medicine industry, including ISCS. We have already known several innovative Israeli startups, but don't have so many chance to learn about the landscape of Israeli regenerative medicine and bioconvergence. It's a great pleasure to have a good opportunity to share information and exchange opinions each other. I hope we have fruitful discussion to promote future collaboration between Israel and Japan today. Thank you for your attention.
野村様、ありがとうございました。 次に、ヤファ、ベンアリ駐日イスラエル大使よりご挨拶申し上げます。イスラエル出張のため、ビデオメッセージにてご了承願います。Ambassador Yafa Ben Ari will share her greetings via video message as she's traveling to Israel.
Shalom. Today, when the whole world is under the terrible impact of the COVID-19 pandemic, in terms of the healthcare, economic crisis, and in many other aspects of our lives, the future of medicine is as relevant as ever. We must always remember to look ahead to the future and ask ourselves how we can make it better. The modern state of Israel succeeded in turning the land of the Bible into a high-tech powerhouse, facing many challenges, lack of resources, security threats, insufficient infrastructure, and food shortages, among others, and made the social and economic developments of Israel and its technological and scientific discoveries an exceptional achievement of the last 7 decades. Israel's claim to fame as the startup country is a result of an ongoing journey of innovation and entrepreneurship. Our advanced nano ecosystem, finding solutions for many global challenges, became very attractive to many multinational companies and countries who are willing to invest and become our partners on the road of growth and prosperity. These days, the positive momentum in the economic relations between Israel and Japan of the last 6 years continues despite the limitations imposed on the development of cross-border commercial activities. Investment from Japan in Israel high-tech actually increased by 20% in 2020. Japanese companies are increasingly interested in Israeli innovation, and the interaction between Japanese and Israeli companies continues to grow. Israel is always willing to share its knowledge. Japanese scientists are excellent in their fields, generating new technologies across many sectors. This event is about enhancing collaboration between Israel and Japan, two countries with amazing capabilities. Each side has different qualities and strengths. We basically complement each other. Working together, I'm sure, we can bring about improvement to lives of mankind. 3 years ago, I had the honor to take part at the signing ceremony of a MOU between FIRM and ISCS. Today, we are taking another step forward to foster collaboration in the fields of regenerative medicine. This time, the Embassy's Economic and Trade Mission, together with the Israel Innovation Authority, organized this virtual event aiming to give the Japanese audience an overview of current situation in Israel, featuring some of the most interesting Israeli startups. Bio-Convergence is an innovative approach that integrates biology with engineering, AI, physics, computer sciences, nanotechnology, material sciences, or advanced genetic engineering to address unmet needs in health and other industries. There is still much more potential to be tapped, and I believe that this important event will have a great contribution in this context, bringing together not only startups, but also researchers to create a better future for us all. I look forward to the presentations and panel discussion. Thank you.
ただいまの動画にて一部に影が入りました旨、お詫び申し上げます。それでは、基調講演に入らせていただきます。本ウェビナーのテーマでもあります、バイオコンバージェンスの推進に努めます、イスラエルイノベーション庁より、カリーナ・ルーベンスタイン氏が当コンセプトにおけます構想をご説明申し上げます。Karina-san from Innovation Authority will share the vision built upon the concept of Bio-Convergence. Karina-san, please kindly proceed.
Good morning, good afternoon, everybody. Thank you, Kanji-san, Daniel-san, the ambassador, and all the participants here. It's really exciting to be here, and thank you for this opportunity. Konnichiwa, everybody. So I'm hoping that you can see my screen now. So the ambassador said rightly that there is a large room for collaboration between Japan and Israel, and I think we second that very much. The fact that we are promoting the future of medicine, as we call it, in Israel, it gives a large playground for collaboration between the countries and the industries and academia and so forth. We're looking at the bioconvergence going through after a long strategic process that we've done at the tipping point now between engineering and biology, and there is a ground for integration between these two sides, between these two worlds and areas to form at the end of the day the next generation or the future of health tech solutions that will face the challenges, the growing challenges of health in the world. And the ambassador already mentioned the areas that we are looking at when we are looking at the integration between engineering and biology. There are many fields that can be adjacent to biology, genetic engineering, and this is the topic of our discussion today: material science, nanotechnology, AI, and so forth. This is the growing industry in the world, and the forecast is looking at $4 trillion of a market potential over the years, all concentrated around human health, but not only agriculture as well, materials, defense, energy, and so forth. So there's a huge potential to grow together in these worlds. We are looking at how do we ultimately form areas that would have a very positive effect on cost-effective drug development, on novel personalized therapeutic solutions, and bio-based solutions beyond healthcare. And if we are looking at the last year of what happened in the world, you can see that these kinds of integrative solutions has really decrease the timelines of developments of technological solutions, bringing the new solutions of health tech immediately to the world. And we see that the last year has made a huge impact on the public awareness of why this is so needed. You can see that in the diagnostic testings, which really eliminated and decreased the timelines until the immediate solution. And so why Israel? Israel is becoming the health tech leader because of pillars and capabilities that we have very strong ones in AI, ICT, and medical excellence, excellent medical research in the academia here. You would later on hear Professor Benjamin Deco, which gives his perspective, but this is only one example of it. We have a strong government engagement is coming from us as a representative of the government here in this discussion, the Israel Innovation Authority, and of course, high quality of medical data. All of that actually makes Israel a very good partner in joining up and performing these kinds of next generation solutions. Later on, there will be speakers here from companies that already, Israeli companies that already started the activity around bioconvergence. So you will hear firsthand their vision and perspective. But as you can see, this is already an industry that is in the advanced steps right now, and some of them are offsprings of the academia in Israel. as you will be hearing later on. We look at bioconvergence, actually, how do we look holistically at specific diseases and transforming healthcare around a specific disease. And this is only an example of companies that actually already evolved in Israel and are active and have and performs a kind of a holistic approach to this to cancer care around early detection, diagnosis and prognosis, treatment decision, and precision treatment. As you can see, all of that gives ultimately a world of solutions around specific disease, but this can be translated to other diseases as well. And so through these kinds of integrated solutions, we actually have a totally different approach to a disease and can shorten the timelines to treatment, ultimately personalize the more precise treatment. Just to give an example of what we are looking at when we talk about bioconvergence, so the next therapeutics discovery tools, whatever relates to microfluidics and lab on chip, for example, the way that more personalized, precise treatment is delivered in a patient's body through nanorobotics, the next generation of CRISPR, more precise editing tools, and the way that we can perform ex vivo trials on 3D printing and cyber tissue engineering through these kinds of technological solutions. So where is us as the Israel Innovation Authority, as a government representative, Israel government representative, where do we plug in? We actually want to create a very good platform for both countries to collaborate, where we can actually create together new growth engines, remove the barriers to entry, and grow the economic value of this new ecosystem together. We are working now on a national program around bioconvergence, where the Israel government would spend large budgets over the next decade to ultimately create a large industry here in Israel of unicorns and many companies in the next decade. And this is a beautiful opportunity to join forces with you, with the Japanese companies and academia, and to develop this vision together. So thank you very much for the time, and I hope this conference will be fruitful for both sides, and we can see future sharing of ideas and innovation together. Thank you.
グリーンさん、Thank you very much. 彼らのご質問はまた Q&A の部屋で、いろいろとお願い申し上げます。では、次の講演へと参ります。イスラエルにおける再生医療での取り組みを、イスラエルステムセル学会会長、ベンジャミン・デケル教授よりご説明申し上げます。Professor Dekel from Israel Stem Cell Society will share his insights and trend of regenerative medicine scenes in Israel. Professor Dekel, please kindly proceed.
Can you hear me?
Yes.
Okay, great. So I want to thank our friends at the Israeli Embassy in Japan, our friends at the Israel Innovation Initiative, and of course the FIRM, our friends for the Forum for Innovative Regenerative Medicine in Japan. I'm a physician scientist, so I speak both languages, the medical and the and the scientific, and I'll share my views as why we're not why regenerative medicine is not reaching the clinic in a way that we would like to see it. We're still within academia, within preclinical efficacy studies, and we want to make it to the clinic. So I want to make a point that we're in the right space for research, right? This is our field, my field of research, the kidney regeneration field. So look, there's about 100,000 people awaiting kidney transplants in the US. 13 people die each day while waiting for a life-saving kidney transplant. And every 14 minutes, someone is in need for a kidney transplant. So, and of course, once you do a kidney transplant, there's need to take lifelong immune suppression. So this is, of course, not ideal. And we are in the right space to rejuvenate organs, to regenerate organs. So at least at the research field, we are in the we're at the optimal situation. And look at this data from the Americans with the registered kidney candidates and the kidney transplant operations. This is just a huge waiting line. I don't know how the situation in Japan is. In Israel, there are about 850 people waiting for a kidney transplant. So that's something we should regenerate, as I said. So regenerative medicine, of course, it's not only stem cells, and it's much wider. It's a combination of cells, biomaterials, and biomolecules that are aimed to rejuvenate and replace damaged or diseased tissues. So the cells are at, we need the cells, of course, but we need the cells to combine with extracellular scaffolds and different genes, different mRNAs. This is a very nice illustration of how we see regenerative medicine at the Israel Stem Cell Society. And of course, it's the cells and the tissue engineering and controlled drug delivery and the immunomodulation and the genetic engineering. And it could be combined, the cells could be combined with molecular therapies and with the biomaterials. So this is the cells, the biomaterials, and the biomolecules combined to regenerate a kidney, a liver, a heart, a thyroid, a parathyroid, whatever, a muscle, a skeletal, the skeleton, whatever you aim to do. And this is from COSO, just illustrations of regenerative medicine. Of course, you can take one cell and put it into the other. This would be allogeneic cell therapeutics. You do have a step of in vitro expansion, or you can, take out a cell and, of course, put in a gene and do what we call ex vivo gene therapy or ex vivo gene cell therapy and correct disease. But mostly when you do ex vivo gene cell therapy, you correct, I would say, rare disease. You correct immune deficiencies, you correct metabolic disease. We're not near regenerating or looking at degenerated organs such as the brain, the liver, the kidney, and we need to go there. And this is my theme of the talk. I sit at the Ministry of Health, the higher committee for cell and gene therapy, and we need to authorize clinical trials in Israel for gene and cellular therapies. So we sit there and we have two opposing themes of thinking. What do we authorize from academia, from industry to the clinic? So we sit there and this is the fast track. We say this, we have some biological sense, okay, for the experiment. The mechanism is a bit obscured. It's not fully delineated, but the treatment is shown to be very safe. It's a safe treatment. So you heard about freedom to treat, right? But, and somebody says there's no preclinical specific model that can predict what will happen in human. So let's move to the clinic. So we don't have a clear biological sense. The mechanism is a bit obscured. It's not fully delineated, but the treatment is fully safe. So we say the patient can is in charge of himself, and we want to move things to the clinic. So there will be a party that actually wants this to go, wants this treatment to go to the clinic. Let's move to the clinic. The other way of thinking is the slow thinking, right? Where the mechanism of action is delineated. Someone has to figure out the biology for years. There's a preclinical efficacy model or preclinical proof of concept on several stringent models, which are close as possible to the clinic. But then if this happens, this might take years and the patient will have to wait. So we're sort of stuck between these two themes. The fast theme, go to the clinic without, completing, let's see what happens in the human being without completing all the to-do lists, or the slow thinking, which is where you have the biology figured out and you have the mechanism of action and you've done it on a small animal model, a large animal model, and the patient will have to wait, but we'll get there. So this is sort of the thinking around these tables where two different parties are pushing towards two different themes of thinking. And of course, I favor this theme, the slow thinking. And this is just from Daniel Kahneman, the Nobelist, the Israel-American Nobelist, who talks about, fast and slow thinking. The fast is unconscious, automatic, everyday decisions, error-prone, and the slow, of course, is to delineate, think hard, go step by step from the proof of concept, from the biology to the proof of concept, do it on 3 animal models, don't be, and then just go to the patient. So I like this sort of fast and slow thinking debate that I wanted to share with you. And this is, the ascending step for evidence. If we want regenerative medicine to be successful in the clinic, we need to work hard in the academia and the preclinical efficacy models and the preclinical efficacy evidence. Usually, you can get projects with have no information available at all. You can get projects that have no information on the cells. You can get projects that have no rationale, no preclinical work, no reports on outcome, no publication. And what we need is this. We need to ascend the steps for evidence. Preclinical work done, solid rationale, protocol available, and outcome published and being visible to the scientific committee. So most of the projects, at least we get, do not have these ascending steps of evidence, and we should really push I would say the academia and the early startups to follow these steps. We need to standardize what is that we require for clinical evidence of regenerative medicine in failing organs. This is critical to be successful in the clinic. And once we do so, who will finance this gap? We get a lot of proof of concept projects, but who will get this gap between the proof of concept to preclinical evidence? And what I say is that I can't share the enthusiasm that everybody has here, because we have this huge gap between the proof of concept to the preclinical evidence, and we need someone to finance this. The academia likes to see the proof of concept. The industry likes to see the clinical evidence, but there's a huge step between the proof of concept to preclinical evidence. I would say, I want to give you an example from our work. This is the kidney, where we generate kidney spheroids from human adult kidneys. These are 3-dimensional kidneys. We put them into mice, and we get these are the kidney spheroids here. We get these beautiful structures, okay? But do they matter? Can you show, this is a proof of concept study. Can you make it can you show that the preclinical POC is undoubtedly solid? And this is hard because you need a small animal, you need a large animal. And I think that if we want to make regenerative medicine a clinical reality, we need to invest on the gap between the proof of concept to preclinical evidence. And until we do that, more and more regenerative medicine projects will fail at higher stages, at the phase 2, phase 3 stages, they're past the phase 1, but they'll fail at the phase 2, phase 3. And what I want to do is try to I'm trying to promote the Israel Society, the Israel Stem Cell Society, to get financing to these proof of concept to preclinical evidence gap. And I think that I would like to also engage FIRM and the Israel Stem Cell Society to get funding for such projects. Thank you.
Thank you very much, Professor Deco, for your frank insights. それでは次に、イスラエル企業3社の発表へと入ります。1社目は細菌感染症への打開策に取り組む BiomX より、ジョナサンさん、アサフさん、ロテムさん3名です。Now we move on to company pitches. BiomX team, please kindly proceed.
Thank you very much. Thank you for having us. We'll let Professor Rotem Solek present first, and then the company will follow.
Thank you, Jonathan. I think, Benjamin, you need to stop sharing your screen so I could share mine. So thank you very much again. It's a pleasure to be here and present our joint endeavor with Jonathan and the team at BiomX. My name is Rotem Solek, and I'm a professor at the Weizmann Institute and a co-founder of BiomX about 6 years ago already. And I'm going to show you some of the studies that we're doing that are at the basis of BiomX's science. And we're studying viruses that infect bacteria, which are called phages. So nearly every bacterium on Earth is being infected by viruses. That comes as a surprise to some of us who are used to thinking of as viruses on viruses that infect humans. But in fact, the most abundant viruses on Earth are viruses that infect bacteria. They're called phages. And like any virus, they identify their target bacteria, they inject their genetic material, and they replicate within the bacterium. And after short time, they will just kill the bacterium and look for other bacteria to kill. And these viruses were discovered about 100 years ago, even more than 100 years ago, and several attempts were done to adapt them as new antibiotics because they are, targeted, basically biological missiles against bacteria. In the initial years, these attempts were not very successful. And one of the reasons is that bacteria have a very successful immune systems to protect themselves from viruses. And you all know the immune system of bacteria. It's called CRISPR-Cas. It gained a Nobel Prize recently. And another aspect of the immune system of bacteria is called restriction enzymes, also got a Nobel Prize when discovered. So CRISPR was discovered relatively late in biology, and still we felt that we don't understand all there is to understand about new immune mechanisms that bacteria encode. So if we want to really develop viruses or phages as therapeutic tools, we need to understand how bacteria protect themselves so we could overcome this protection. So if we understand bacterial immunity, it will lead to better design of phage therapies. And also from another applicative aspect, we realized that if we discover additional defense systems that were not known before, in previous discoveries of such systems, it led to revolutions in biology, like restriction enzymes, CRISPR, and other immune systems. So in the lab, we have been developing in the past few years a systematic way to discover new immune systems in bacteria. This study or this series of studies is combined of a very large-scale computational learning AI-style discovery engine that scans tens of thousands of microbial genomes, hundreds of millions of genes, and predicts which genes will be involved in defense against viruses. And then we take an experimental approach and put these genes into model bacteria and test whether they indeed provide defense against viruses, against phages. And over the past few years, we were able to discover a large number of new defense systems, each of them encoding a very exciting new kind of biology. And overall, so far in the past 3 years, we discovered more than 15 new bacterial immune systems that were not known before. In addition to these discoveries, we discovered that phages can communicate. It's another, profound knowledge on phages that was missing from the body of knowledge of humanity. We found that some phages can use small molecules to communicate and make group decisions. And all these findings, which kind of revolutionized the field of, phage biology and bacterial immune systems, are really driving the innovation of BiomX. So we are in constant contact with BiomX, and the information flow that we derive, is shared with BiomX and then drives their commercial innovation, which you'll immediately now hear about from Jonathan. Thank you very much.
Thank you, Rotem. I'll move to share with you the presentation of BiomX. So again, I think we're very honored to be here, and I think humbled to have scientific founders such as Rotem Sorek, truly one of the leaders in the field. To give you all a little bit of a snapshot of the company. So we are actually the only company out there that is in the clinic testing patients and volunteers using phage on chronic indications. And again, based on all the technology that Rotem presented. Over the last 5 and more years of existence of the company, we've invested heavily in building the platform. The company is probably the phage company with the most numbers of employees. We're 110, probably raised the most amount of capital. And the investment has produced this Bolt platform, which encompasses, I think, themes that we'll talk about of bioconvergence. Also, as you'll see through the presentation, our connections and interactions and relationships with Japan run very deep, from investors to some of our fundamental science. So what we can do in BiomX is actually move very quickly using the Bolt platform. We can actually initiate a project and get a readout in patients within 12 to 18 months, which is actually unparalleled time if you compare it to small molecule or antibodies that usually will take 5 to 6 years to get to that point. We took a decision to do in-house manufacturing, and we can now produce 50 to 100 clinical grade phage every single year, which we've jumped tenfold in the last years based on convergence of bioinformatics, computational biology, and a lot of the synthetic biology approaches that we deploy. If you look at the pipeline, we have a very broad pipeline. We already have positive phase 1 data in acne, which is the lead program, and we're now in the midst of a phase 2. We have a positive phase 1 PK study in our IBD PSC program that I'll elaborate more. Very interesting program with technology coming out of Japan, out of Keio University. And basically for BiomX, this is going to be a very exciting year and a half, as we'll have 4 phase 2 readouts that read in the next 15 months. So we have data in acne phase 2, phase 2 in IBD, a phase 2 in cystic fibrosis, and a phase 2 in atopic dermatitis. On top of it, there are a few collaborations. We have a collaboration with an international cosmetic company around our asset in acne. That company has asked to remain unnamed for competitive reasons, and the SEC has approved it. We have 2 collaborations around our computational platform. Think about that in a company of 110 employees, 10% of our workforce are actually computational biologists. Because of the importance of understanding our targets and how phage interact with bacteria, which is basically utilizing all the science that Professor Sorek has mentioned before. And that is also the basis for 2 additional collaborations, one with Johnson & Johnson and the other one with Boehringer Ingelheim. The company to date has raised $120 million. The first 2 rounds were private rounds that were led by OrbiMed, Takeda from Japan, and Johnson & Johnson. For those who are familiar, we also have SBI, who's a very prominent Japanese investor. The SoftBank joined the 2 private rounds. And in October 2019, we went public in New York, and that brought in another $60 million. With that, just to give you a snapshot of the company, and then I'll do a bit of a deeper dive. So as you can see, there are 5 programs in the pipeline, with many of them having a substantial readout in the very near future. So acne is a program that we've already reported positive phase 1. So we've actually shown that administration of phage on the skin reduces bacterial load with statistical significance. So we have a proof of mechanism. And we've just embarked on the phase 2 with the results that will read out in the end of this year. And here, the question that we're asking ourselves is whether we can see not only reduction of the target bacteria, but also improvement in the appearance of the skin. That's exciting, and we'll read out end of this year. Inflammatory bowel disease and PSC. So inflammatory bowel disease is, of course, Crohn's, colitis, and diseases associated with it. And PSC is an orphan liver disease, primary sclerosing cholangitis. These programs share the same target. We're extremely excited to announce a positive phase 1A. So this was the first time in the world that phage were delivered orally under an IND with the FDA clearance. And we've seen a very supportive PK profile as well as the therapeutic window. We will launch the phase 1B, phase 2A end of this year and expect results first half of next year. I will go into more details because these are extremely exciting programs that originate from Keio University, from Professor Honda Sensei and Professor Kanai Sensei, who have published these findings in prominent journals. Interesting point is that Professor Kanai Sensei is actually Abe Shinzo's personal physician because of his expertise in this field. And this has been a long and very productive relationship and a very warm relationship that we're very proud of. Cystic fibrosis, that is a program that we've launched not too long ago. We do expect to treat patients at the end of this year and have results by the end of this year. So that's testimony of the platform and our capability of moving extremely fast. The program was launched in November of last year, and we already are in clinical grade manufacturing, gearing up for a clinical study. As I said, this is probably 18 months until we get a data readout. What will be in a normal small molecule, probably a 5 to 6 years process. Atopic dermatitis, another interesting program, a huge unmet need. And we are advised by the dean of the School of Medicine at Keio University, Amagai Sensei, and it is a very relevant target with a market of $5 billion that are expected to go to $20 billion in the next few years. Again, we are expecting phase 2 results in the first half of 2022. And there is an earlier program in colorectal cancer that we'll have to read out in the next few months. So to make everything work together, and that's the capability of the Bolt platform, as you can see here, there is a reason the company is 110 employees at a relatively early stage, and that's because of the complexity of what we do. We are pursuing, at times, novel targets, so different bacteria that were not known to be relevant in such indication. We need to understand more about what phage we find, and as what Professor Solek mentioned, understand all their defense system, what are the vulnerabilities of the bacteria, what are the vulnerabilities of the phage, and we basically built a pipeline which is both robotic and computational to screen out hundreds of phage and hundreds of bacterial targets until we found phage that are worth pursuing forward. We have quite a lot of capabilities in synthetic biology, and this also comes from one of our scientific founders, Tim Liu from MIT, one of the leaders in synthetic biology in the world. And at times we will engineer the phage to add capabilities and payloads when we need to. And at times we'll also engineer the host, so the bacteria that we grow the phage on them, to make them more suitable for manufacturing. Then starts a long process of cocktail optimization with iterations after iteration. Some of it is in silico, some of it is in vitro, some of it is in vivo. Once we have a cocktail that looks good, a process that usually takes 1 year to 2 years, but recently we have cut it down to a few months because of moving to a theme which is a lot more computational, a lot more based on metagenomic, which is, I think, we'll all be interested today to talk about Bio-Convergence. Because of that and the addition of all these computational pipelines that have actually been endorsed by the FDA, we can cut this timeline dramatically, move to manufacturing, do everything in-house, and then start dosing patients when we're ready to do so. To give you a sense, so two of our most exciting programs, the first one is an inflammatory bowel disease. As I said, this comes from the work of Professor Honda Sensei at Keio University with a joint appointment at the Riken Institute. And what Professor Honda has published is actually work that he takes bacteria and samples from patients. So he takes whether it's fecal samples or saliva samples from inflammatory bowel disease patients and implants them in germ-free mice. And at Keio University, there is a big setup to work on in mice that have no germs whatsoever, and you can control which bacteria you're entering and which one are in the model. And here, what we can see is that in some of the mice that he put samples from the patient, he saw inflammation in the liver. In the gut. This is inflammation that looks like the human disease. So when you put bacteria from the patients in mice, he actually saw something that looks like the human condition. After a lot of work, he actually understood that there's a single bacteria which is driving all this inflammation, which is a bacteria called Klebsiella pneumoniae. He then took that bacteria to another mouse model and showed that it exacerbated clinical symptoms. So what's interesting, and that's what got him the publication at Science, is the discovery that a single bacteria was actually responsible for all the inflammation that was seen in these mouse models. He then teamed up with a professor at the Broad Institute, and they analyzed patient samples, and they actually saw that patients have a lot more of this bacteria than healthy volunteers. At this point, one of our scientific founders from the Weizmann Institute, Ranan Inab, is also on this paper and collaborates with Professor Honda Sensei. And we got involved. I flew to Japan a couple of times. We've established a relationship and signed an agreement with Keio University. We've actually got the bacteria. We've replicated all the data and gathered 500 fecal samples from patients across the world. And we do see that patients have a lot more of the bacteria compared to healthy volunteers. So that makes it a really interesting target. And I will add that recently, a study by another company called Ceres in the microbiome is also indicating that in these patients, if you can reduce the Klebsiella bacteria, you're going to see an improvement in the patient's condition. So again, a very large unmet need. Many of these patients don't actually respond to some of the new drugs out there. And truly an opportunity to have an intervention which is safe and could be very substantial in these patients' lives. Now, I think what got us even more excited is that separately, a separate group at Keio University, led by Professor Amagai Sensei, who, as I mentioned, is actually Abe Shinzo's personal physician, was looking into this orphan liver disease, PFC. And they again took fecal samples from the patients and implanted them in germ-free mice. And what they saw is in some of the mice, they actually saw inflammation, you can see here, in the liver. And they were asking themselves, why are we putting the bacteria in the gut and getting inflammation in the liver? That does not make sense unless the bacteria was somehow moving. They were actually creating a hole in the gut lining. And this is actually what turned out to be. They actually found bacteria circulating inside the mouse, with bacteria literally causing a leaky gut, so physically poking a hole in the gut of these mice. And after a lot of work, they understood that this phenomena and this behavior is actually driven by a single bacteria. Interestingly, it was Klebsiella pneumoniae again, same target in the IBD. Again, this group has reached out to us because of our previous relationship with Keio University, and we've licensed the technology and the rights to this target. This was published in Nature Microbiology. And we are working together, sponsoring research at Keio University, and actually replicated all the data. And we're extremely excited about the target as there's more and more clinical evidence that supports the role of this bacteria in disease. The fact that reducing this bacteria is going to have a significant effect. This is an extremely antibiotic-resistant bacteria, so antibiotics is not an option. And on top of it, we know that antibiotics will kill some of the good bacteria that are important for these patients. So you actually have no other choice but to use a phage. By the way, what's also interesting, if you think about the two different labs in two separate...
Jonathan, I will ask you to wrap up in 30
question.
seconds. Thank you.
Okay, no problem. So I'll just wrap up and show, just to give you the highlight of where we are. We have the acne program that will read out in a phase 2 end of this year, the IBD PSC program that will read out in the first half of next year, CF that reads out in this year, and atopic dermatitis that reads out first half of next year. And again, for us, the relationship with Japan and the role of Bio-Convergence is definitely crucial. So I thank all of you.
Thank you very much to BiomX for the input. そして2社目は、がん治療に取り組むナノゴーストより、ヨナタンさん、マーセイさん2名から発表させていただきます。また運営からの、おすすめなのですが、今の Zoom のレイアウトの方で、スピーカービューモードにしていただきますと、より一層見やすくいただけるかと存じます。The Nano-Ghost team, please kindly proceed. Thank you.
Okay. Thank you, everybody. I'm Jonathan, the CEO of Nano-Ghost. Thank you for having us. I want to let Professor Marcel Makhlouf to present the technology, and then I will add a few words about the business side. So please, Marcel. Marcel, we can't hear you.
Okay, so thank you very much, and thank you for the invitation to present our technology, and of course the company, as Jonathan outlined. This is a company that actually I will present first myself. I'm a professor at the Technion, and I was working on the technology that I will present shortly for the last 10 years. So the company itself is based on this technology, and it was established a year and a half ago. And as I will show later on, the technology is a very unique platform that can target different type of solid tumor and therefore deliver different payload. The technology itself relies on a very strong IP approved in the US, Europe, India, and pending in China. And we have a lot of preclinical studies that we already performed, but now going on into the company with the scale-up process, and of course, trying to interest partners with different payload that can be loaded on this Nano-Ghost. So as Jonathan mentioned, I am a founder. Jonathan is the CEO. We have here also Dr. Osnat Bohana Kashtan, who is the VP R&D, and the company include several researchers. The technology is, the name of the technology is Nano-Ghost. The name of the company is the Nano-Ghost. The technology is based on mesenchymal stem cells. This is adult stem cells that are very unique and are known to home to different type of solid tumors, and also protect the tumor from the immune system, allowing the tumor to grow and invade and metastasize. So the idea behind this technology is to identify only the part that knows how to interact with the tumor, and in this case is the cellular membrane, which has all the receptors and ligand that interact with the tumor niche. And then we took these cells and we wanted to control the technology by actually preparing only a mimicking vesicle. And we therefore empty the cell from its content, from all its cytoplasmic content, and then left only with the membrane itself. We developed a technology in which we can downsize this membrane to a nanoscale, and then we can fill this nanovesicle platform with different payload, ranging from small molecule to a large protein, nucleic acid, and I will give some example of the efficacy of this system. So that's how the Nano-Ghost look like. There are a round spheric vesicle, around 200 nanometer, but we can control also the size. They resemble much like a liposome, with a very huge difference that the membrane already include all the targeting and interacting compound and receptor needed to identify the tumor niche. They also express all the markers, especially the unique one of MSC, like CD29, 90, CD44. We already know that these Nano-Ghosts interact with different tumor cells, and they interact by or endocytosis or by fusion with the cell membrane, where we found them mostly in the cytoplasm and where they can dump their payload. And also we saw some indication that part of this Nano-Ghost goes and interact with the nuclei, which opened the door for non-viral transfection that I will show later on. Another important feature, these Nano-Ghosts do not interact much with the lysosome, and therefore they escape the lysosomal compartment and can save their cargo, which needs to be deployed in the cytoplasm. We have several studies already performed. This is one of the latest one, not yet published, soon to be published, where we show that the Nano-Ghosts not only interact with the tumors, but also can pass different different barriers, like the BBB, the blood-brain barrier. Here we can see the Nano-Ghost in red interacting with U87 cells immediately after 30 minutes of incubation. And we can see also we develop a BBB model in vitro that they can pass this BBB in a short time, 30 minutes. When we did intracranial GBM mouse model and injected the Nano-Ghost IV, we saw accumulation of the Nano-Ghost in the tumor niche, but not in the normal brain tissue, nor in the infiltrating organ. In another model that we did of glioma cancer stem cells, where we see lesion of tumors in the brain, we saw that our cancer stem cells that are labeled here in GFP are co-localized with Nano-Ghost in red. Again, these Nano-Ghosts were injected IV to these animals. This is only another example of what can we do with the Nano-Ghost. We also can load them with RNAi as well as RNA. That's what we are working currently in the company. In this example, we loaded the Nano-Ghost with anti-MIR RNAi that inhibits MIR221. And in this case, we can see the cell, the Nano-Ghost here labeled in green, the MIR are labeled in red. And again, using this Nano-Ghost anti-MIR, placing them on the cells, we can see that we need less than the amount that is needed with lipofectamine to transfect the cells. And if we increase this amount of MIR, we can have a higher effect than lipofectamine. These studies was also done in vivo, showing that we can deliver the MIR or RNAi in the Nano-Ghost. As I mentioned, the Nano-Ghosts interact with the nuclei, therefore it opened the door to try and see if we can use them as a non-viral vector. In this case, we loaded the Nano-Ghosts with the cDNA that encode for PAX, the C-terminus of MMP. This is an inhibitor or anti-angiogenic and apoptotic drug that kill cancer cells. The Nano-Ghosts were loaded with this cDNA via electroporation, simple electroporation at the end of the process, placed on the cells. And we saw that the different tumor cells are transfected via this Nano-Ghost, leading to the expression of PEX and of course the death of the cells immediately after transfection. We did also in vivo studies in mouse with non-small cell lung carcinoma, inoculated in the lung. And in this case, the Nano-Ghosts were loaded with the cDNA that express PEX, the similar that I show in the in vitro study. And we can see here that IV injection of this Nano-Ghost again reached the lung, leading to 50% inhibition in the metastatic lesion occurring in the lung. Another model that we have tried was prostate cancer inoculated subcutaneously. In this case, the Nano-Ghost was loaded with a protein, the soluble TREL, which is known as an apoptotic drug, but also stopped in phase 3 clinical studies because it is unstable protein, and therefore you need to give a lot of this protein, a huge amount, in order to get any result. Therefore, we took this protein, loaded it in the Nano-Ghost, injected again IV to mice bearing this prostate tumor. What we can see here that first of all, the Nano-Ghost arrived to the tumor. The Nano-Ghosts are labeled here in red, and they cover all the bulk of the tumor, which is very unique. Usually drug delivery system, if they arrive to the tumor niche, they reside at the outer surface, release their cargo, the drug, and then the drug need to diffuse. In this case, the Nano-Ghost so much resemble the MSC that the tumor themselves, the tumor cells uptake or bring the vesicle inside the tumor, disperse them all over the niche. And what we also saw that one injection with 50-fold less of the drug led to 85% inhibition of the tumor growth. Again, only one injection. So I hope that I convinced you. We have a lot of other data that we can share. I hope that I convinced you that we have here a unique delivery platform that can target itself to the tumor niche and therefore can deliver different payload. And now I will give Jonathan to explain about what can we do. Yes. and connection that we are looking for. Thank you.
Thank you. I know we don't have enough time, but I would like to add a few words about from the business side. So as Professor Marcel showed you, the Nano-Ghost technology is a wide platform for delivery, for targeting treatment for cancer. And this technology could be implemented for many types of drug for cancer and also for inflammation. So as a platform company, our strategy is to develop our own plan, our own drug, 2 to 3 projects that are developed by our own resources. But because this is a wide platform, we are also exploring the opportunity of collaboration with pharma companies. So if you, as a pharma company, and you have a drug, or develop a drug, or construct for cancer, and you are facing with a delivery issue, so I want to invite you to contact us, and we can explore the opportunity together to find and develop, to combine our platform and your technology and to develop a great drug. So thank you very much again. So, and I'm waiting for you and enjoy the webinar. Thank you.
Thank you very much for your input, Nano-Ghost team. 皆様にもしよろしければ、ぜひQ&A の機能をご活用いただき、登壇企業へのご質問などいただけましたら、幸いです。3社目は、視力回復、人工網膜に取り組むナノレティーナより、ヤエルさん、ラニさん、2名より発表させていただきます。Nano-Retina team, please kindly proceed.
Thank you, everyone, for the opportunity to show Nano-Retina and its journey. With me here is Professor Yael Hanin from Tel Aviv University, which will be presenting some of the slides. See, just one second. Okay, so Nano-Retina has set a mission to restore vision to patients blinded by degenerative disease. And this is something that we said in the beginning that we want, this is mainly the mission of the company. It's established in 2009, located in Herzliya, Israel. We have a team of 20 multidisciplinary employees, basically emphasizing the Bio-Convergence subject. We have biologists and engineers from almost every discipline you can imagine, making this a possibility. The technology that we will present shortly in the next slides basically is enabled by global partners that we have from almost everywhere you would like in the world. The microelectronics is developed and manufactured in Israel. We have MEMS components that is performed or manufactured, designed in Israel and manufactured in Europe and North America. We have a glass ceiling enabling the biocompatibility of our implant inside the eye. We have a Japanese partner in Tokyo and Hiroshima enabling this to be possible, and finalizing in Europe, and electro-optics in Europe and North America as well. The company right now is at the stage of clinical trials, and we already implanted 4 patients with this, and this is right in the middle, but this is very exciting times for us at this stage. Right, I'll hand over to Professor Yael Hanin to explain a little bit about the technology. Yael, we cannot hear you.
Sorry. The very first thing is to understand the location of the device and the manner by which it's supposed to operate. So the target the device is addressing is the visual system. And as you can see here in this short video, you can see a cross-section of the eye and the operation of the healthy eye. In the next slide, you will see a video showing the fundamental idea of what happens when the retina goes through a degeneration process. In such a process, the photoreception that is typically performed by the inner layer of the photoreceptors that you can see on the right is no longer active. And that fundamental process leads to visual loss, which could be either partial or complete. So the operation of the device is meant to replace the photoreceptors, and it really builds on the fact that the rest of the layers are still functional and useful. So on the right now you can see a device, a schematic of a device that is implanted on the retina with spikes needles that penetrate through the retina. And these needles are MEMS needles that are designed to interface with the remaining layers of the retina and transfer electrical signals that elicit electrical activity of the retina, which then goes to the brain to achieve visual perception. So this is the very fundamentals. Now in the next slide, you can see really how the whole system is built. It's actually built of 2 components. There's the device that you've seen, which is a tiny implant, a few millimeter implant that is implanted in the eye on the retina, on the epiretina side. And this implant is communicating basically with eyeglasses. So the NR600 implant is inside the eye. The NR600 eyeglasses are mounted on the face, and they communicate with the implant, delivering both energy and information, which is needed for the operation of the device. The visual information itself is delivered through the eyeglasses. And this slide is really, the title of this slide could be also Bio-Convergence. This is a real, I think, a very, very nice example of hardcore bioconvergence, where you can see where a medical problem recruits expertise in MEMS, in ASIC design, in packaging, in optics, and a lot of different technologies. And as Lani mentioned, the technology builds on activity from many different parts of the world, where everything converges into the implant, as you've seen before. So a big part of the development was the 3-dimensional needles that you can see in the picture, and these needles facilitate part of the anchoring of the device to the retina. They're also specifically designed to deliver the right kind of safe stimulation that is needed in order to elicit the neuronal response in the retina. Everything else has to be optimized, minimized in order to reduce the interaction between the device and the biological system. So this is looking a little bit into more details into this device, both back and front views. And at the back, or actually the surface that interfaces with the retina, you can see those silicon needles with very special coatings that had to be optimized to achieve the best interfacing. And on the upper side, you can see through a glass ceiling, which have to be, of course, hermetically sealed for many years, basically protecting both photovoltaic systems and ASIC elements and the many other elements that were carefully integrated into this very, very tiny and compact system. The eyeglasses are external, so in some sense were easier to develop, but they also were carefully designed to enable both the energy transfer and the control of the implant. And they have, we really benefited from the collaborations with many international leaders in order to achieve such complex engineering task. And the last slide for my part is this slide, which basically shows a very simplistic view of vision delivered with an implant. So what you can see on the left is an original image, a video image, and then what does it mean to take that particular image and transfer it to specific discrete pixels and black and white pixels. So it's very clear that reducing the number of pixels or increasing the number of pixels have huge impact on the kind of information that the patient can ultimately observe, perceive. And our main effort right now is really to optimize not just the number of the pixels, which at the moment is 600 electrodes, but also the detailed information which is delivered during the stimulation. And this is a very big part of the development process of these devices.
Thank you, Yael. I would like to speak a little bit about the market potential and about the disease we're treating. So in terms of, there are two major diseases that we are aiming, targeting, the AMD, which is age-related macular disease, which is actually the most substantial market, and retinitis pigmentosa, both of which are degenerative disease and which patients are born and they can see in the beginning, but gradually as their lives continue, in retinitis pigmentosa at the very early stage of their lives, around 20, 30, or 40 years, they start to lose their sight until they cannot see completely. AMD is usually happens when the patients are relatively older, at around 60s and 70s. And most of the market is there. At the moment, we are starting our clinical trials with retinitis pigmentosa, and later on we will continue with the AMD patients. Regarding the competitive landscape, so there are groups all around the world trying to develop such implants that will enable vision. Not a lot of them are actual companies. A lot of the effort is done in academic groups around the world. Regarding the companies that are working on these, there's the most leading ones are the Second Sight, which have the Argus 2 and the Orion. Argus 2 implanted such implants in over 400 patients and have now stopped implanting these devices. They have very low resolution, as you could see in the slide that Professor Hanin presented. And then they are starting to implant these implants in the cortex, visual cortex in the brain. So this is in the early stages. Another company that is working on this is a French company called Prima. They're called Pixium. They have their product Prima, which is developing also implants, and they are a good competition for us. Doing similar, the idea is the same, but the engineering or the implant itself is a little bit different. So we are both running along the path and trying to make this possible. Regarding the clinical status, so company is now aiming at completing a 20-patient study to obtain a CE mark, which should be done somewhere around 2022. There are 6 leading clinical centers around the world, 2 in Belgium, 2 in Italy, and 2 in Israel. For patients who have been completely blind for a few years, our participants are already implanted with the device and are in follow-up, and we are doing a lot of research with these patients in order to improve the technology and understand how to improve resolution, and also understand what could be done for even better resolution in the future. We've proven safety on these patients and efficacy, but this is just the beginning. We understand there's a lot more to do. This is just a quote from Professor Stalmans from University of Leuven, who has performed the all 4 implantations so far. And he says he was very excited, obviously, to see patients who have not seen for over 5 years and already started to understand and see objects and images and catch items. And again, this is a patient that has been completely in the dark for over 5 years. This is an amazing, amazing thing to be part of this project and to see this. We're hoping to see more and more of these in the coming months. Regarding the clinical regulatory strategy, so we're, as I said before, we are aiming at the CE mark towards 2022, and then moving on to FDA. In parallel, we'll start working on AMD patients, and this will be further along the road for us. Regarding the leadership, so it's we have, as I said, we are about 20 employees in the company. We have myself as the CEO and VP R&D, Professor Yael Hanin, who is with us and helping us with the research programs, and David Rigler, who is the VP R&D. And most important, we have scientific advisors, global scientific advisors from around the world helping with this, both from the surgical part and the surgery part, and also the ophthalmologic and improved vision for the patients around the world. So this is briefly us. Thank you very much.
Thank you very much for your input, Nano-Retina team. 3社へのご質問は経済部にても改めて受けたまわります。The Economic Commission will also relay your questions to these 3 companies at the end of the opportunity. では、パネル講演に入らせていただきます。Now we move on to the panel discussion. こちらは私ども経済部の代表、ダニエル・コルバーが進行役を務めます。Daniel Culver, heading the Israeli Economic and Trade Mission to Japan, will moderate the panel discussion. Daniel-san, please proceed.
あやさん、どうもありがとうございます。Thank you very much. Hope you can hear me well. Good. 皆さん、こんにちは。イスラエル大使館の経済講師、ダニエル・コルバです。よろしくお願いします。Shalom to all the friends and participants in Israel and in Japan. We're very happy to have you all with us today for this virtual event. This is the first of a series of events touching this subject, and our objective is to see how we can promote collaborations between Israel and Japan. So over the next 30 minutes, I will have the pleasure to talk with our distinguished panelists precisely about this subject. I will first like to ask our panelists to briefly introduce themselves, starting with Suzuki-san from FIRM, please.
Hi. Okay. Thank you very much for this opportunity for FIRM. And my name is Suzuki, and I'm a participant for the activity of the firm as a vice chair and also the member of the board. And I spent a long time to make the industrial advocacy activities through the firm. And firm, one of the firm's mission is to as a name of the firm, our main objective is to promote the industrialization of the regenerative medicine, which include cell and gene therapies. And today I'm very happy to participate this panel. Thank you very much. Thank you.
Thank you very much, Suzuki-san. Next, can I ask Mr. Arya Kramer from Takeda, Israel?
Konnichiwa, everybody. Konnichiwa. Happy to be here. Thank you very much for the invitation. My name is Arya Kramer. I'm the founder and the managing director of the affiliate of Takeda in Israel. Founded 8 years ago, and now currently the biggest pharmaceutical company originated from Japan, who is actually working very strongly in Israel. Happy to be here.
Thank you very much. Ms. Karina Rubinstein, you presented before, but please take a short time to introduce yourself as well.
Yes, so thank you again, Daniel-san, and everybody here. Happy to be here. Konnichiwa. I mentioned already in my presentation, but I represent the Israel Innovation Authority, which is the government agency for promoting the economy of Israel through investment and innovation. We have been doing so for many years, promoting collaborations with the international markets, and Japan is of a paramount interest to us to promote these kinds of collaborations. Specifically, the Innovation Authority is leading the Bio-Convergence national program. And I think, and you saw the presentations here and all the speakers, there's so much areas that we can collaborate in, a lot of activities that can be done together. And I'm so hopeful that we can do that. And thanks again for this opportunity. I think it's a great kickstart for this kind of activity.
Thank you very much, Karina. Professor Dekel, last but not least.
Thank you, Danielle, and thank you for organizing this great event. So I'm a professor at Tel Aviv University. I'm a pediatric nephrologist in my clinical hat, and a stem cell biologist in my research hat, and I head the Israel Stem Cell Society, which looks to collaborate with FIRM very intensely towards bringing regenerative medicines to patients.
Thank you very much. Okay, so Suzuki-san, my first question is for you, as the firm's mission is to pave the way for real breakthroughs in regenerative medicine by pursuing broad industry-led partnerships with governments, with universities, with private sector, to promote the commercialization and application of new regenerative medicine technologies. I would like to ask you about first, if you can briefly talk about challenges to achieving these goals, and also about opportunities for collaborations with Israel.
Okay, thank you very much. The thing is, first of all, as you know, FIRM has already made a MOU with Israel Stem Cell Society, the Professor Benjamin, the Dekel, and through this kind of relations, firm are very happy to participate these occasions led by Embassy of Israel, and to know the regulatory framework or market of the Israeli regenerative medicine market is one of the first step for us. And then for the Israeli companies or the researchers who like to expand their activity into the Japanese market. And we are quite happy to work together with all those kinds of people. And in order to achieve one of the mission of the firm to promote the industrializations of the regenerative medicine here in Japan, is through the collaborative work with Israeli companies or non-Japanese companies, and we introduce their activity into Japanese market, and that kind of the discussions is quite important. So therefore, through this occasions today, most of the audience comes from Japanese societies. Therefore, I think today's three presentations, all of them are quite innovative the modalities of their technologies, and I'm very interested in that kind of activities, those kinds of activities. So therefore, I believe my colleague here in Japan also have a very interested in that kind of towards the three companies' activities, and also the speeches of Professor Dickel, I believe. Thank you very much.
Thank you very much. Yes, indeed, we all listened to the presentations by the Israeli companies and was impressed by the ongoing cooperations with Japan. So we would like to see more of that. Mr. Kramer, a question to you. Takeda's operation in Israel, you founded Takeda Israel as well. Could you tell us about Takeda's activities in Israel, and especially in relations to innovation and to regenerative medicine?
Thank you very much. Yes, so Takeda was actually affiliate, was founded in 2014 as part of the global transformation that Takeda actually took in order to become a more specialty focused pharmaceutical company. There was an understanding that the Israeli market is a strong market when it comes to innovation, both from the perspective of ideas that are coming from the academia, but also a strong place in order to make business, to provide sophisticated cutting-edge technologies, pharmaceutical technologies to the Israeli health population because of the fact of a strong appreciation to innovation. So as part of this kind of transformation, Takeda understood that Israel is a market that needs to be represented locally. So this was actually the rationale to Takeda operation. We started working on specific therapeutic areas. Now we are operating in GI, gastroenterology, oncology, neurosciences, rare diseases, and plasma-derived therapies. As part of our journey to deliver more unique technologies to GI specialists, we actually launched 2 years ago, actually the first stem cell therapy which was approved in the world. The name of the product is Darvastrucel. It's actually coming from allogeneic stem cells from Oedipus-derived donors and treating complicated perianal fistulas, which is some type of complication of Crohn's disease, speaking back to what BiomX actually mentioned. And this was done in a very strong participation of the Israeli society, meaning that Israel was one of the significant contributors to the pivotal studies that took place in all over Europe. Actually, 20% of the patients were actually coming from Israel. And this was actually the first approval in the world for advanced therapy, which involved regenerative medicine. I'm happy to say that also when it comes to the implementation, Israel is one of the largest country when it comes to the actual experience with this technology, and we are contributing together with the local HCPs community in order to better understand the mechanism and what are the experience which is gained behind this kind of technology. When it comes to innovation, it's not only doing business with Israel. Takeda also established in 2014, together with the partnership, strong partnership of the Israel Authority, represented here by my friend Karina, the local bio-incubator, which actually is a platform for introducing new technologies or new ideas which are coming from the Israeli academia into a platform that could have the framework to establish the proof of concept implementation and follow-ups to advance this kind of companies into a massive or broader impact. Currently to date, this bio-incubator, together managed by the Israel Authority and together with additional companies, is actually a host for almost 30 companies, which is actually being raised there, being supported by the leadership of our partners, the pharma companies who are actually having the ability to generate the support and the required direction towards successful clinical development. And Takeda is actually looking for additional opportunities to expand this kind of activities in Israel. So very strong appreciation, very strong satisfaction from the collaboration with the Israeli Authority. And we are looking forward to continue this kind of partnerships.
And so how would you say would be the advantage of Takeda over its competitors in terms of scouting for technologies or enhancing its research capabilities? Takeda's presence in Israel, that is.
Yes, so my presence here as I would say the site manager of Takeda in Israel is that to be a very, I would say, close address for any kind of ideas or discussions, both for the local governmental agencies, but also for potential researchers or potential scientific community who would like to get into connection with Takeda. This is a very strong, at the end, Israel is a small country and everybody is actually knowing everybody. So this gives you, I would say, better visibility in order to be exposed to new ideas, and as a gateway within Takeda, I have the ability to, to make sure that this kind of ideas will get the right attention within the organization. Further, as part of the head of the site of Takeda, I'm actually taking part of significant agencies or trade associations which actually are working very strongly in order to shape the local Israeli arena when it comes to become more friendly into innovation, working closely with the government. We are part of I'm a member of the board of Pharma Israel, which is the local trade association for all the multinational pharmaceutical organizations, and we are working very closely with the government in order to make sure that this kind of innovation will continue to thrive within the organization or within the country. So the fact that there is a representation of the multinational in Israel contributes significantly to the advancement of this kind of collaboration compared to some kind of discussion only abroad.
That's for sure. And we do have about 400 multinationals having their R&D centers in Israel. And that's definitely helping to the innovation ecosystem. Now, Arya-san, you mentioned the incubator, and I wanted to ask Karina if you could elaborate about the new call for proposals that would be relevant for Japanese companies.
Yes, so I just wanted to mention to Arya, maybe you can say a few words about how Takeda joined back then to the incubator. It was really on the fly kind of a decision that they took, and not so, I think it's, not so common for Japanese companies in this magnitude to make such decisions, but they were really fast in it. and it's proven itself this kind of a magnificent activity over the years. They've been now active for 7 years already, and you saw BiomX back before talking about they sprang out of this incubator. They were provided with all the guidance, the platform, whatever they needed, beyond the funding, of course, from day one. So I think this is the right platform to go about it when you are investing in innovation and you want to expose yourself to open innovation. This is the right way to go about it. We have the Israel Innovation Authority actually supports nowadays around more than 20 incubators across the state of Israel. This is the flagship program of the Innovation Authority, has been running for 3 decades already, and has sprung hundreds of startups from scratch, from actually zero. A lot of them are commercialized from the academia in Israel. Some of them are IP brought from abroad. This is a way to go about it as well. And we actually fund 85% of project budgets within this incubators. This is a way for us to lower the risk for companies that would like to expose themselves to open innovation from day one. And now, as you mentioned, Daniel, we do have an open call. It's a beautiful opportunity to establish 3 new incubators in Israel and be exposed to the ecosystem, thriving ecosystem in Israel, be part of our Bio-Convergence vision, and join up with other multinationals, venture capitalists, medical centers from Israel. Benjamin Deckel, Professor Deckel can mention, how Sheba can plug into these kinds of activities, but that's not the only example. And of course, the academia in Israel work with startups, entrepreneurs, and so on, with our, very high investments in these startups. So I call for you all to join up with this opportunity.
Yeah, certainly, and anyone who would like to hear more about it, we have all the details, and we'll be happy to provide more information. Certainly recommended. Professor Deckel, over to you, and thank you very much for your insightful presentation. I'd like to ask you, since we're talking about presenting to the Japanese audience what is happening in Israel, if you could highlight what are the strengths of the Israeli researchers and startups in regenerative medicine, and what are the challenges?
Thank you, Daniel. So of course, as everybody mentioned, innovation is part of our sort of society, and there are many innovations in the field of stem cell biology and regenerative medicine in Israel. And the human resource, I would say, is exceptionally sophisticated and well educated. So both innovation and human resource are, I would say, our engines for development and in both academia and industry.
Mm-hmm. Now, the Israeli Stem Cell Society signed, was mentioned before also by Suzuki-san and the ambassador about 3 years ago, an MOU with FIRM. So could you tell us what were you hoping to achieve with the MOU? What type of partnerships would you like to pursue?
First of all, I wanted to see Suzuki face to face, and my Japanese colleagues face to face. And we didn't know about COVID when we signed the MOU. We hope to sort of generate a delegation that would participate in face-to-face discussions, which is, I would say, much more productive and could really, generate teams of specific projects on different fields of regenerative medicine that we can find mutual interest in. So, of course, that would, some sort of a conference delegation, that sort of, and that would might have lead to joint funding. And I think, as I mentioned before, we need to take the innovation and show that it works, and show that things work on funding mechanisms. And I think that both FIRM and Israel Stem Cell Society can generate, with Israel Innovation Authority, could generate specific funding mechanisms around certain fields that we could, certain projects that could be very promising at the innovation stage, and take it to the stage where we can show that it works, as I said, and gather a lot of evidence prior to starting clinical trials. So I would hope for both conferences and face-to-face contact, and of course joint funding towards joint goals.
Yeah, certainly. We will talk about COVID-19 in a minute. Before that, could you give us a brief overview in terms of regulation? What is happening in Israel? Is it a factor that it's facilitating the progress of regenerative medicine? And how are the international?
Of course there is. I mean, you can't go through the local IRB now. With regenerative medicine therapies, you can't. I mean, you have to go to the higher committee of the Ministry of the Israel Ministry of Health, which I shared my sort of my views from the inner talks there. And the thing is that we follow FDA and EMA guidelines. So that's in terms of authorizing this sort of regenerative medicines. But still, there's always a conflict in terms of which aspects can you be more, I would say, more relaxed on, and which should be do not authorize. The authorization, non-authorization has to be, I would say, more formulated around the world with regenerative medicine. I mean, the Americans just made the fast track for regenerative medicines, and I think that's good. But we're still sort of debating how live medicines, which is most of regenerative medicines are live medicines. They're not dead medicines. They're live medicines. Should be regulated and should be taken to the clinic. But as I said, mostly it's with the FDA and EMA guidelines that we adhere to.
May I?
Thank you. Please.
Yes, thank you, Professor. Your explanation is quite interesting for us, and for the Japanese the players who has intentions to work together with the Israeli startup companies who has or researchers who has a very innovative technologies in the regenerative medicine or cell and gene therapy market. At that time, the Japanese players like to know the what kind of the R&D activity has been made in Israel, and also what kind of regulatory framework, including a non-clinical, and also including a safety test, and also the POC type of the concept. Those kind of the steps. We like to know what kind of process has been made in Israel. Of course, the Israel has a very close relationship with say US or Europe, and you have the very good relationship with FDA and the EMA. We understand. In addition to that, we expect the Israeli government to work much closer to the Japanese PMDA, and if there are limited gap between the Japan and the Japanese regulation and the Israeli regulations, for the Japanese players, it is easier to proceed the project into the Japanese market. So therefore, in future, through the MOU activity between FIRM and the Stem Cell Society, we make a certain level of the communications to know each other about the regulatory framework and also the market difference, and that kind of the step is very important. I believe my colleague Max Nomura, he will take initiatives to work together with you in very near future. That is one of the important steps for the both parties or both countries. Thank you.
Daniel, may I add something to Mr. Suzuki's comments? Just to add that we are in discussions, long discussions as part of the national program in Bio-Convergence with the Health Ministry to promote advanced, innovative regulation framework around these areas, specifically advanced therapeutics and so forth. And they are working with us together on developing such a framework. So I think this is in the right timing right now. In relevance to Japan, we would like very much to take these inputs, and somehow plug them into these kinds of discussions that we have with the Health Ministry, together with Professor Bdeko.
Great, good. So we already have an action item from this session. Very good. Arya, back to you. I wanted to ask you, since this is we're talking about the future of medicine, and so we're talking about the future, and obviously we'd like to know about trends. If you can share with us, if you have some thoughts about what are some of the current trends in terms of bioconvergence or regenerative medicine that you see coming up in the near future?
I think that both aspects are actually answering, I would say, the focus of our representing the industry. The focus is to, I would say, provide or generate a much more sophisticated solution, cutting edge technology, which actually answers the specific need of the specific patient. Personalized medicine is the new king, and both regenerative medicine and bioconvergent solution could actually address this. I think that you can see, if you can see from Takeda example, now there is a very strong focus on mRNA, if you saw, so the collaboration between Anima Biotech to Takeda in order to generate specific small molecules, which is actually a new modality, new small molecules which actually affects or controls mRNA signaling. It's a unique platform, and this is actually was found in Israel with a small, very modest deal of $1.2 billion. So there is a lot of focus on personal medicine. This is why we believe that Israel could be a very strong platform for generating this kind of collaboration. Also, as Professor Deckel mentioned, there is a very strong focus in the pharmaceutical industry now, with a better understanding of the genome revolution, on focus on making solutions for genetic and metabolism elements or diseases, which could also be affected by the new modalities, which are actually demonstrated also by BiomX. So this is actually put the main focus of companies like us in generating this kind of collaboration in Israel.
Very interesting. And the follow-up questions for you, and maybe actually for everyone, all the panelists. We haven't talked about it almost yet, but we have a global pandemic going on. And the question is, how is that going to impact regenerative medicine, in your opinion? For the better or for the worse? I mean, for example, we saw one challenge is that it is making it difficult for international collaborations because there are no conferences and no travels, etc. But I'll be happy to hear your thoughts on that, starting with you, Arya.
I can think that the experience that we saw from the pandemic is actually putting, I would say, additional thoughts to all the regulators and also to companies like us in making sure that maybe, and just commenting to the dilemma that Professor Deckel also mentioned, what should be, I would say, the more expedited process in order to approve or to evaluate the efficacy and safety of new molecules, new technologies. We saw that there was an exception in this sense when it comes to the solution generated by the vaccine companies. And this is actually a generation, generates, I would say, some kind of, maybe we should think differently. And you can see some kind of early science when it comes to the regulatory agencies where they are saying, maybe we should collaborate in a much broader sense together as part of mutual collaboration. You can see this kind of examples we call Project Orbis of the FDA, actually summing up all, I would say, the free world, the Western world, the agencies like Canada, Switzerland, Australia, also Israel and Japan, in order to try to evaluate technologies in parallel in order to shorten the time for approvals. So I think that this could be a very, very strong quantum leap in the process that we could consider post-COVID. We need also to benefit and to appreciate the remote interactions that we are generating. As Israeli, I don't say that this is actually could replace totally I would say, the personal engagement, but we don't see significant interruption when it comes to interacting with the HCPs, physicians, thought leaders over the digital tools that we are actually using now, speaking directly between Israel and Tokyo and Osaka, which is amazing, and saving a lot of resources. So I think that we will see in the upcoming future significant change in the ways that we are working in this sense.
Okay, very good. Hopefully for the better. We saw Israel reacted very swiftly when it comes to vaccinations, and here in Japan, it's covered very widely in the media. And I myself went to Israel to get vaccinated. Actually, I'm very happy about that. Any other feedbacks and replies to this question before we wrap up the panel? Karina.
I would like to add, and I think I mentioned it already in my presentation as well, that it helped speed up times. We saw that in achieving advanced solutions in very short timing, and I think this is a process. My thinking is that this is a process that will stay there, hopefully, and we cannot go back now, to the old system, the old-fashioned doing things. This pandemic, hopefully, we can see the light at the end of the tunnel already, but we don't know what the future lies ahead, God forbid. And I think that we should be ready. And I think the readiness is within the technology and the innovation. And if we spend our attention, we put our attention there, we should be ready for whatever lies next. So my thinking is that we should act now and should be adopting to these agile kinds of processes that we already saw, and were happening naturally, in a sense, last year, and still in ongoing processes. We are very much hopeful that we can be one of the drivers of change in this aspect.
Yeah.
Suzuki-san.
Yes. When we talking about the COVID-19, at the time, even here, say Israeli population is smaller. However, the percentage of the vaccinations has been made is quite high. The percentage is quite high. We understand. So we Japanese people say envy your country has made the very good the actions. However, Japanese people also expect the very near future vaccination should be made sufficiently. However, we are now, even in Tokyo, we are under the very strict, not locking, but it's a difficult restrictions for the activities. However, through the digital technologies, we can make this kind of communications, and it is this kind of with Corona or after Corona or post-Corona, the way of working style should be changed. So therefore, communication style also should be changed. So we can say we can overcome the distance, and the time-wise, time difference also that we can overcome. So therefore, I hope we can make a much more say communication and making a project in near future. And also we Japanese players also they are very interested in getting say some support from the Israeli government. the hope say working together in the near future.
Certainly. Very well. Professor Decker, did you want to add anything?
Yeah, I just want to re-emphasize what Arya and Karina and Suzuki said about standardization across boundaries, you know, of the how we may take regenerative medicine to the clinic and, you know, do it good and fast as possible, you know, combine both the, as I said, the POCs and go further. But I do want to mention that, you know, the COVID is a great example of let the science talk. You know, when Katin Kariko and Drew Weissman published their paper in 2005 in Immunity, nobody thought that this would change the world. You know, they generated the first modified mRNA. I'm not sure people heard about these two scientists here in the crowd. I don't know, but their science led to the discoveries by Pfizer, you know, and the German group and Moderna. Okay, so this tells us again that we should let science talk and let you know, because science is the game changer. It changed, of course, COVID outcome, and we should adhere to talking science, and that is a great lesson also from this pandemic.
I think, Benny, you know, every child knows now what mRNA is. So, you know, at least we've done that. This has been done, you know, up until now. And maybe this is the biggest
but I'm
change.
not sure they know what is. I'm not sure they know what is modified mRNA. They know what mRNA is, but no one is paying attention to what is modified mRNA and how modified mRNA was invented, actually. And I think that was, you know, it's such a beautiful lesson to all of us, people, you know, the people who do the work quietly and sustainably, actually change the outcome of this world. You know, I didn't know about these two scientists until up till now, and I would, you know, I would.
Modified mRNA is the next message we should convey. You know?
Yeah,
right.
Everybody's now into
this. For the next session.
Yeah.
So I feel like we could have talked
medicines with modified mRNAs.
I hope that we will not need additional
we could have continued. Gentlemen, ladies, we have to conclude. Unfortunately, we could have talked for longer. It's very interesting, but we are running late, and that's my fault. So I'd like to thank you all again and for the interesting discussion. And back to you, Kenji-san. Thank you.
Thank you very much to all panelists for your insights. パネリストの皆様ありがとうございました。最後に協力機関であります、JETROより理事、曽根一郎様からご挨拶を受けてまいります。We are grateful to have Mr. Ichiro Sone, Executive Vice President of JETRO, to share with us his thoughts in closing. 曽根様よろしくお願いいたします。
Well, thank you. Hello, everyone. I'm Ichiro Sone, Executive Vice President of JETRO. I would like to thank everyone from the Israeli side for joining this wonderful webinar today. In the pandemic of COVID-19, Israel is drawing global attention to its pioneering efforts, as discussed at the panel, especially vaccination at the fastest pace in the world. Even in Japan, such prevention efforts are reported on TV news, and even non-business persons have become familiar with the name Israel. Last year, Japanese companies invested more than $1.1 billion in Israeli companies, a record high. In addition, the number of Japanese companies based in Israel has increased to 90. And the economic relationship between the two countries has become more active than ever, even in this corona pandemic. I have great expectation for Bio-Convergence as a booster to develop our relationships further. Bio-Convergence is a new field. Therefore, I believe it can be realized with Israel's unique ideas and Japan's engineering capabilities based on our deep mutual understanding and discussions on the topic. Finally, I would like to express my respect and gratitude to everyone who participated today. Her Excellency Ben Ali, Ambassador of Israel to Japan, and members of the IIA and FIRM, thank you very much. Toda Rabba.
曽根様、どうもありがとうございました。後日、皆様へアンケートを送付させていただきます。お手数ながらお時間よろしくお願い申し上げます。また、各企業とのお時間をご要望の方は、私ども経済までご一報お願い申し上げます。Lastly, for further inquiries, please kindly reach us, your Israeli Economic and Trade Mission. イスラエル企業及び研究機関との連携、取り組みへのご支援、改めて感謝申し上げます。今後ともウェビナーのご視聴、イスラエル企業との個別会議などよろしくお願いいたします。本ウェビナー、これにて終了とさせていただきます。We appreciate your support for making bilateral collaboration in commerce and research moving forward. We look forward to having all of you again for more events to come. This will conclude today's webinar session. Thank you very much.
Thank you very much.
Thank you.
Thank you very much.
Bye-bye.
Thank you. Bye-bye.
Thank you very much.